We are discussing the mechanism of transposition, and in this segment, we will discuss the mechanism of the transposition, how its cell performs this function, which is very important. Now, transposons, they are sometimes called the jumping genes, and the DNA doesn't always leave one place for another. This is one fact. And when DNA leaves the place, it can be non replicative, non transposition, which is like cut and paste. As we cut and paste the file, similarly, the transposons are cut from one place and are pasted to another. Now, both strands of original DNA, they move together from one place to another without replicating. Just consider that when we move or when we cut and paste the file in our computer, what happens? The original is there and the whole file is moved itself per se to another place. Now, transposition frequently involves DNA replication. That is, the DNA replicates and one copy remains at original sites, as I have said earlier, and new copy inserts at the new site. Replicative transposition, copy and paste, that is the process which is the replicative transposition. That's the one way of the mechanism. In the first step, the two plasmids, they fuse, phage replication forms a co-integrate completed through the pair of TN3 copies. Now, TN3 copies, that's the mobile gene in the prokaryotes, which is known as that. In this diagram, this thing is just illustrated that there are transposon, which is in the low brown, and the green is the target DNA, a target place. And TNPA fusion, it occurs and co-integrates into a DNA molecule where it further goes back to transposon, is liberated, and the DNA PR resolution is again formed. So you can see that the copies remain there, but it is cut and paste and it moves to another place in the TN3. Now, the detailed DNA3 transposition that has been demonstrated in this figure, where you can see that the transposon and the target are there. In the first step, what happened? That the DNA is nicked. As I told earlier, that nick means that it is DNA strand is separated, is cut at the places which are observed or which are just illustrated in this figure. Then the step 2 is the joining the loose ends, and further the step 3, the replication of the transposon, which is performed, and the step 4 is the completion of the replication, where the replication or the duplication, it is completed in this case. Another way mechanism is non-replicative transposition. That starts with the same two first steps as in the replicative transposition. New nicks occur at the narrow mark, and then nicks liberate donor plasmid minus the transposon. As we have seen that the plasmid and the donor transposon, they are liberated. They are formed from there, as is shown in this, that there is a nick occurs, DNA at arrow, and further there is a double-stranded DNA made, and further the gaps which occur between the DNA strand, they are filled together. Now, filling gaps and sealing nicks completes target plasmid and its new transposon, they are generated.